Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Hope you all fine. Dear students, this lecture is for grade 10, subject chemistry. In this lecture, we will discuss the remaining topics of chapter number 13, that is biochemistry. This is my 23rd lecture, and I am your lecturer, Mrs. Saira Jamal. In this lecture, I'm going to talk about the classification of carbohydrates. Carbohydrates are classified into three main classes. The first one is monosaccharide, which are also called simple sugars, oligosaccharides, which are composed of 2 to 9 sugar units, polysaccharides, they are composed of 10 to 1000 sugar units. About the monosaccharide, I explained in my previous lecture. Today I'm going to talk about the second class of carbohydrates, that is about the oligosaccharides. Dear students, as I told you that oligosaccharides are composed of 2 to 9 molecules of monosaccharides or simple sugars or sugar units. Oligosaccharides, same like monosaccharides, are white crystalline solids. They are soluble in water. They are sweet in taste. Oligosaccharides are further classified into different classes, and this classification is on the basis of monosaccharide units or sugar unit which produced upon hydrolysis. It means that oligosaccharides, when react with water, upon hydrolysis they give different sugar unit, different number of sugar units. Those number of sugar units decide about the type of oligosaccharide. The first type is disaccharide, which give double sugars or double sugar unit upon hydrolysis. It is the sugar formed when two monosaccharides, simple sugars, are joined by glycosidic linkage. Two sugar units combine together to form a carbohydrate or an oligosaccharide, which is called disaccharide. And which type of bond link those two sugar units together? That type of bond is called glycosidic linkage. Glycosidic linkage is a type of linkage which hold two sugar units together. They upon hydrolysis yield two sugar units. As I told you, when they react with water, disaccharide will react with water, so it reacts with water and will give two sugar units. And as I told you, what is hydrolysis? Hydrolysis is the chemical reaction or the chemical breakdown of a compound due to reaction with water. So disaccharide when react with water, they will give two sugar units or two monosaccharide units. Examples of disaccharide are, you can see the formula of sucrose C12H22O11, and when sucrose react with water, it will hydrolyze or it will breaks up into two sugar units. One is glucose and the second monosaccharide unit is fructose. We have learned it in the previous lecture that glucose and fructose both are the monosaccharides and they are hexoses, like they are 6 carbon containing monosaccharides. Glucose is an aldehyde and fructose is a ketone. Glucose contains aldehydic functional group and fructose contains ketonic functional group. Second example is of lactose. You can see the formula of sucrose, lactose and maltose. They are same, and they all of these three examples, they react with water, and upon reaction with water they breaks down into two sugar units or two monosaccharide units. In the first case for sucrose, glucose and fructose, and in second case lactose will after hydrolysis will give glucose and galactose, and maltose after hydrolysis will give two glucose units. So all these three upon hydrolysis give two sugar units or two monosaccharide units, so they are called disaccharides. So these three are the important examples of disaccharides. Second class of oligosaccharide is trisaccharides. Tri from the name indicated that they are composed of three monosaccharide units with two glycosidic bonds, and the bond which join these three monosaccharide units are called the glycosidic linkage. And how many glycosidic linkage will be there? Two glycosidic linkages will be there, or bonds will be there. They yield three sugar units upon hydrolysis. Example A is raffinose. Raffinose, when dissolved in water, it will hydrolyze and after hydrolysis it will give us 3 sugar units or 3 monosaccharide units. Then we have another class is tetrasaccharide, which will give 4 sugar units. Pentasaccharide will give 5 sugar units, and so on and so forth, depending upon the number of sugar units which these oligosaccharide gives upon hydrolysis. Then we call them disaccharide, trisaccharide, tetrasaccharide, pentasaccharide, so on and so forth. Coming toward the third class of carbohydrate, that is polysaccharides. From the name indicated, poly means many and saccharide means monosaccharide units or sugar units. Carbohydrates which upon hydrolysis form 100 to 1000 units of simple sugars are called polysaccharides. They are amorphous solid. The disaccharide and or oligosaccharide and monosaccharides are crystalline solids, while the polysaccharides are not crystalline in nature. They are called amorphous solids. They are tasteless. Oligosaccharide and monosaccharides are sweet in taste and are soluble in water, but polysaccharides are tasteless and insoluble in water. They are non-reducing in nature. Reducing, I told you, those substances which by themselves are oxidized because they loses proton and will act as a reducing agent. So polysaccharides, they don't have any hydrogen to lose because already the monosaccharide they loses hydrogen atom and then they linked together through glycosidic linkage, so no hydrogen is present at carbon number 1 to donate it and will act as a reducing agent. So they are non-reducing in nature because they are not giving any proton to any other molecule. Examples are starch, cellulose, and glycogen. They are the examples of polysaccharides. Now we have self-assessment exercise 13.1. Classify sucrose, lactose, and maltose as mono, di, and trisaccharides. So you know that sucrose, lactose, and maltose, these three are disaccharides because I told you sucrose is made up of two sugar units, that is glucose and fructose. Lactose is made up of glucose and galactose, while maltose is made up of two sugar units. So sucrose, lactose, and maltose all are disaccharides. Since sucrose consists of two monomers, therefore sucrose is a disaccharide, which we call a table sugar. Table sugar is called sucrose. Cane sugar or sugar itself. Sucrose is a disaccharide of glucose and fructose. Maltose is a disaccharide of glucose and glucose. And lactose is a disaccharide of glucose and galactose. So lactose is equal to glucose plus galactose. All these examples are of disaccharides. Question number 2, is galactose a monosaccharide? Yeah, galactose is a monosaccharide because it cannot be hydrolyzed and consists of only one unit. So those carbohydrates which are composed of only one unit, one sugar unit, are called the monosaccharide. So galactose is a monosaccharide, but raffinose after hydrolysis gave us 3 sugar units. So raffinose is a trisaccharide. Self-assessment exercise 13.2 give 3 examples of monosaccharide, disaccharide, and 1 example of trisaccharide. So 3 examples of monosaccharides are glucose, mannose, galactose, fructose. There are 4 examples of monosaccharide. For disaccharide are maltose, lactose, and sucrose. And for trisaccharide, in your book is raffinose. But other examples are maltotriose, maltotriose, this one is igarotriose and isomaltotriose. These are other examples of the trisaccharide. And polysaccharide are cellulose, glycogen, and starch are the examples of polysaccharides. List sources of sucrose. This is the second question. Sucrose is present, obtained in sugar cane, sugar beet, honey, and fruits. Maltose is obtained from cereal. It is also found in beverages, beer, cereal, pasta, and potatoes. And lactose is the main sugar. It is present in milk and all the dairy products. Coming toward the topic sources of carbohydrates, carbohydrates are the most abundant class of carbon-containing compounds. First one, monosaccharide. Monosaccharides such as glucose, fructose, and galactose are obtained from fruits, vegetables, and cereals. They are also present in honey. So all the sweet fruits and vegetables, they contain monosaccharide. Honey also contain fructose in it, which is a monosaccharide. Disaccharides such as sucrose is obtained in sugar cane, sugar beet, and fruits. Maltose is found in cereals. Lactose is main sugar in milk and dairy products. Then polysaccharide, that is example is cellulose and starch. Cellulose is obtained from plants. For example, cotton. Cotton is pure cellulose. Starch is present in cereals, wheat, barley, rice, maize, potato, sweet potato, etc. We can obtain starch from all these vegetables. What are the uses of carbohydrates? Carbohydrates store energy in both plants and animals. In plants, carbohydrates are converted into starch, and in animals, carbohydrates are converted into glycogen, where they provide energy. 1 gram of glucose gave 15.6 kilojoule energy. Only 1 gram of glucose will give 15.6 kilojoule energy. They serve as food source for most organisms. Carbohydrates serve as structural material for plants, like the plant body, like the cellulose, means the stems, branches, all are made up of cellulose. So carbohydrates serve as a structural material for plant. Cellulose in the human diet is referred as fiber. It is found in bran, whole meat bread, fruit and vegetables. We cannot digest it, but it is very important for us because they are fibers, and fibers are required for the proper digestion of food. And although they are not digested by the stomach, but they help in digestion. So it helps the muscles of your intestine to move food efficiently through the digestive tract. It absorbs and carries away toxic chemicals in food. There are two functions of the fiber. The first one is they help in the digestion of food, and the second one is they kill all the toxic chemicals in our body. Would otherwise harm us. It also helps us in lowering cholesterol and regulate blood pressure. By eating the fibers, we can control the cholesterol level and the regulate blood pressure. Sucrose is used as a common table sugar. Common table sugar is sucrose. So glucose is stored in animal muscle and liver cells in the form of glycogen. I told you the extra glucose in animal is stored in the form of glycogen, and in plant it is stored in the form of cellulose and starch. Glycogen serves as long-term energy reservoir. It can be converted back to glucose when needed for energy. Plants store excess energy as starch. Starch is used to make rectified spirit by fermentation process. Rectified spirit we are using it in our labs in the form of ethanol. Ethanol is called rectified spirit. Starch is converted into dextrin, which is used as an adhesive for stamp and wallpaper glue because starch is sticky. Cows, cattle, goats, deer, sheep, and termites derive nutrition from cellulose. The cellulose, like the plant stems and their branches, are food for termites, and the leaves also food for goats, cattle, and all the sheep, deer, etc. We use cellulose in the form of wood for heating, housing, and furniture. Wood is also used to make paper and wood pulp. Cellulose fiber of cotton is used to make rayon and cellulose acetate, which are used in textile energy for making clothes. You can see in this image that these acetates are used to make the rayon, and these rayons are then used to make clothes. And the second one is acetate, so we can use this acetate to make clothes of it. The second next topic is about the nucleic acid. What are nucleic acid? Nucleic acid are nitrogen containing compounds. They are long chain molecules made up of nucleotides, and each nucleotide consists of three components: nitrogenous base, number two pentose sugar or 5 carbon sugar, number three phosphate group. They are vital component of our life. They are found in all living organisms. They were first obtained from the first cells of infected wound by Swiss biochemist 100 years ago. You can see in this image, this is an image of the three components which are present in nucleotide. One was I told you the phosphate group. This is PO4 phosphate group. Then ribose sugar. This five carbon or five member ring structure is called ribose sugar, and also it contains nitrogenous bases. Nitrogenous bases are two types, purine and pyrimidine. Purine are composed of adenine and guanine, while pyrimidine is composed of cytosine, thiamine, or instead of thiamine, sometimes uracil is present. You can see that purines and pyrimidine differences are purine are double ring structure, while pyrimidine is single ring structures. So phosphate group, ribose sugar, and nitrogenous base. These three are the important component of nucleotide. They combine together to form nucleotide. And then many nucleotides combine together to form the nucleoside. Another we can say many nucleotides combine together to form the DNA helical structure. There are two kinds of nucleic acid, deoxyribonucleic acid, which is called DNA, and ribonucleic acid, which is called RNA. You can see in this image the structure of DNA, which is double-stranded structure, and this double-stranded structure is composed of nitrogenous bases. Nitrogenous bases are in a specific code, like in adenine and like thymine will be in pair, and cytosine and guanine will be in pair. These are called genetic codes because these pairs are very important for the production of new organisms. So this must be in pair, adenine with thymine and cytosine with guanine. And this is the double helical structure of DNA, and all the strands are joined together through hydrogen bonds. This is the structure of ribonucleic acid, which is single-stranded structure, and nitrogenous bases are present, but the structure is single-stranded. Let us discuss the first one, deoxyribonucleic acid, which is also called DNA. Structure of DNA was discovered by J. Watson and Francis Crick in 1953. DNA exists in the form of two strand or double strand twisted around each other in a spiral formation called double helix. Each chain or strand is made up of deoxyribose sugar, phosphate unit, and nitrogenous base. These are the three units of a nucleotide. The strands are held together by hydrogen bonds. The order of the base pair in a strand is a code that stores information which is used to produce proteins. The key to the ability of DNA to store genetic information and to pass on from generation to generation is its double-stranded structure. What is the function of DNA? DNA can store and transmit all the genetic information needed to build organism. For instance, in human beings, the single fertilized egg cell carries information for making legs, hands, head, liver, heart, and kidney. All parts are formed by this DNA. Then after DNA, the next one is ribonucleic acid, which is also called RNA. Structure of RNA, I told it is single form, single stranded structure. It is made up of ribose sugar. The deoxyribonucleic acid or DNA is made up of deoxyribose sugar, while the RNA is made up of a sugar called ribose sugar, which contains hydrogen with it. Ribose sugar, phosphate unit, and nitrogenous bases. So function of RNA is RNA synthesized by DNA to transmit the genetic information. RNA is responsible for directing synthesis of new proteins. RNA receives, reads, decodes, and uses genetic information from DNA to synthesize proteins. So the main function of RNA is that RNA synthesize protein and they collect information from DNA. DNA will give them a specific genetic code. Like I told you, the genetic code that which nitrogenous base come in front. So after that, you will like RNA receive, read, decodes, and uses genetic information from DNA to synthesize new proteins. Because I told you that the codes are very important. Genetic codes are important. So this was all about the DNA and RNA, and also classification of carbohydrates. We discussed the monosaccharide in my previous lecture, and in this lecture we discussed about the other two classes of carbohydrates, their sources of carbohydrates, their uses, and then we discussed the next topic, DNA and RNA, like their structure and their function and their double-stranded structure, and the nitrogenous bases which are present in nucleic acid, all about them. So inshallah the next topic we will discuss in the next lecture. Till then, take care. Allah.